Hi, I'm Dr. Bijal Shah. I'm here at the first annual iwCAR-T meeting in Miami, Florida. We just finished up a brilliant session on CAR T-cell therapy and acute lymphoblastic leukemia. I'm joined by my fellow panelists, Dr. Shah from Medical College of Wisconsin, Dr. Gardner from Seattle Children's, and Dr. Roddie from University College London. So why don't we start with you, Dr. Shah? Tell me a little bit about your talk today and what we should take home.
Yeah, so our group at the Medical College of Wisconsin was looking at a bispecific construct that is a tandem sort of CAR T cell that can target both CD20 and CD19. We think that's an exciting way specifically for non-Hodgkin lymphoma because both of those targets are sort of considered to be present on all B-cell non-Hodgkin lymphomas. The other thing that we did that was unique is we used a point-of-care manufacturing system, the Clinimax Prodigy device, which allowed us to make CAR T cells in a GMP environment without the same rigor and requirements of a full GMP facility. And so what we were able to show in this phase 1 study is that you can make CAR T cells with the Clinimax Prodigy. We had 100% success in patients who were CAR naive. And I think we have exciting results for patients who achieved the goal dose of 2.5 million CAR T cells per kilogram. We had 9 out of 11 patients achieve a complete response. Future studies are indicated, but we think we have something that's worth further investigation.
Dr. Shah, can you tell me a little bit about how many patients fell out during the process of manufacture? And then as a second follow-up question, what you were seeing in terms of CD19 antigen loss using this novel CAR?
Yeah, so we did have 4 patients who had prior exposure to a commercial CAR T cell product. They all had received a CD19 CAR T cell. And when we tried to apheresis those patients and remanufacture our CAR T cell product, 4 of those patients failed to make the goal dose. And 3 of those patients subsequently did receive treatment off protocol, but none of them had a clinical response. So clearly an area that we need to investigate more is how to treat patients after they've had a prior CAR T cell product. In terms of relapse, what we saw, which was I think interesting, is we didn't see downregulation of CD19 or CD20 as a dominant mechanism of relapse. And in fact, we saw high levels of CAR T persistence in several patients, suggesting there is antigen stimulation, but clearly there's something else going on that is making the CAR T cells ineffective in terms of killing the tumor. So there's definitely areas that we need to investigate more to better understand the mechanics of this bispecific construct.
Absolutely. It sounds like this might be a good segue into acute lymphoblastic leukemia.
It is, and we're hoping to start a clinical trial with our children's hospital, which is connected to our adult hospital in a phase 1 children and young adult population with refractory B-cell acute lymphoblastic leukemia.
Great. Dr. Gardner, would you like to share some of the highlights from your talk?
Yeah, I think building on Dr. Shah's talk, so for pediatric B-ALL, we have had a lot of excitement now for a number of years with CAR T-cell therapy. And as that time has gone on, we have really been humbled by the lack of durable remissions. So of all the patients that get into remission from CAR T-cells, a large number of them, up to 50%, will ultimately recur. And so now I think what's most exciting in the field is the development of new strategies. So how can we make the CAR T cells persist longer? How can we develop novel CAR constructs that may be superior to what we initially did? And building on Dr. Shah's talk as well, this idea of dual targeting. So we know that a lot of patients will have antigen escape. They will downregulate or lose expression of CD19 or CD22. So how can we develop new treatments that have multiple antigen targets available to hopefully improve the durability of those remissions?
That's incredible. When you do see loss of B-cell aplasia, or when you do start to see loss of CAR T-cell in the circulation, you mentioned that there are some strategies that you've employed at Seattle Children's to try and for lack of a better word, re-engineer the CAR-T expansion?
Yeah, so a few strategies that we've been working on to improve persistence have been developing adjuvant therapies to go along with the CAR T cells, as well as developing CAR constructs that are from fully human single chain FVs. The idea with the fully human scFV is that you will not develop rejection responses and you can have more durable upfront persistence. And one of the findings that we had in our initial studies of ALL was those patients that had low numbers of CD19 positive cells going in had a smaller expansion and were more likely to lose persistence of their CAR T cells. And we and others have shown that if you lose your CAR T cells early, you're going to recur. And so we took a group of patients that had low antigen load, less than 15% disease in their bone marrow, and engineered T cells that expressed a CD19 protein. And our early experience has shown that when we infuse these TAPCs to patients after they receive their initial CAR T cell infusion, that we can enhance their persistence by episodically stimulating their CAR T cells in vivo. And this has been done with really little side effects, so it's very tolerable and really early promising results.
Great. Dr. Roddie, tell us a little about your talk.
So the focus of my talk today was really adult acute lymphoblastic leukemia, which is really an unmet medical need. Once patients relapse, there's very little in the way of good therapeutic options. And there's very little, in fact, available to certainly patients in the UK in terms of CAR T cells for this indication. So at our centre, we've devised a series of clinical studies specifically for this group. And today I focused on the iwCAR-19 study, which is probably our most exciting ALL study at the moment. And effectively, it's distinguished a little bit from other studies and other CAR constructs in the sense that the binding moiety, so that's the single chain FV that targets the CD19, it behaves slightly differently. It has a different binding kinetic to CD19, and so what that means is that our CAR-Ts are in contact with tumour for a lesser time period than other conventional CAR-T cells. And so we were initially concerned that maybe having less time in contact with tumour would result in a lower tumour kill and perhaps lower expansion of the cell product. But in fact, the opposite has been the case. So what we've found using this construct is that in fact the expansion is excellent and mirrors what's seen with other conventional CARs. But what's exciting about it is the fact that the persistence of this product seems to be really quite significant. And we've got patients on both the pediatric and adult studies who've got expression of their CAR beyond 12 months and up to sort of 2 and 3 years. The other exciting thing about this CAR is perhaps by virtue of the fact that it's in contact with its target for a lesser time period, it means that it secretes less in the way of cytokine per interaction with target. And what does that mean? Well, ultimately, if you have less cytokines produced, we know that a lot of the toxicities that are intolerable to patients, and particularly to older and more fragile patients with adult leukemias, we feel that having lesser cytokines perhaps results in lesser toxicity. And in both the pediatric and in the adult study, looking at this CAR, we report negligible grade 3 or more toxicities in these patients. So if you're looking for a product that could be applicable broadly across our older patient cohorts, particularly with lots of comorbidities and poor performance status, I think this is probably a very exciting potential product for those patients.
I couldn't agree with you more. Exciting was exactly the word that I was going to use. It's very cool technology. One of the things that you didn't have the opportunity to talk as much about, and I want to give you some space and time to talk about it, is the concept of the polyfunctional or polysecretory T cell. Do you find that dynamic is a little bit different with this design, or is this something that you think is applicable across CAR-Ts? I just wanted to give you a chance to talk about that.
Well, yeah, that, as I say, whenever I was actually giving the talk, I feel that my definition of what is an acceptable CAR-T to give to a patient at the moment is very binary, in the sense that I have a set of release criteria and my team generate cells with a certain viability, with a certain level of expression of CAR on their surface that are sterile, so on the surface of things look safe to give. But of course you give what you perceive to be the same product to 10 patients patients with sort of similar disease profiles, and in fact you may get 10 similar response patterns. And the fact that I can't define the potential of my therapy before I give it is something that gives me sleepless nights. And so it was with great interest I came across the paper by John Rossi and his team, looking really at this. It's a sort of a novel platform called the isoplexis, where you have the capability to perhaps assign a sort of a polyfunctionality index or a measure of potency in a sort of a preclinical way after you've made your product, to try and help you define whether that product is going to be able to mount a response in vivo or not. Because up to the current time, it's a bit of a black box and we don't really know what to expect. So it's very early days for that for us. We've literally only just got our hands on this piece of equipment and we're getting to grips with how to sort of, run the samples. But I think going forward, it's going to be hugely helpful. We'll define what patients we think are going to respond. We'll be able to introduce manufacturing methodologies that can perhaps sort of make patients with poor polyfunctionality more effective downstream in vivo. So I think there's a lot of potential in this piece of technology. So excited to talk about it at the next iwCAR-T, I guess.
Or maybe ASH?
Or, well, no, that's too early.
Too early?
Yeah, too early.
Very good. I want to take this opportunity to thank my panelists. Again, Dr. Shah, Dr. Gardner, Dr. Roddie, all phenomenal presentations. I think we're all in an era of plenty, and we need it. I think that there are significant unmet needs across the board, whether we're talking about preventing CD19 antigen loss, getting CAR T cells to patients faster, whether we're talking about improving the activity and persistence of CAR Ts. Again, whether we're talking about identifying a platform, a novel platform, a novel design for enhancing the efficacy without this signature of toxicity, we're getting one step closer, I think, to that finish line. And again, I'm just humbled to be a part of it. So, thank you.